In this video, we will continue our discussion where we have stopped in our previous video. So before going into the comparison of SARS-CoV-2 spike protein glycosylation in different cell system, let us first understand some behavior of the SARS-CoV-2 virus. So this is the SARS-CoV-2 virus. We can say uh, major domains. Uh, Here it can be seen that it starts with from the 5' end. It starts with the ORF1A 1B. After that there is a spike protein domain, and then 3A 3B. Then there is another structural protein, that is envelope protein, membrane protein. Then other domain 6 7A 7B 8A 8B 9B, and then there is a nucleocapsid domain. The important thing that we need to know in this is that major mutations in the SARS-CoV-2 occurs in the spike protein domain. Within the spike protein domain, as it is, this is the expanded view of the spike protein domain. It is the expanded view of the spike protein. Within spike protein, the major mutation that occurs that is in the receptor binding domain, and this is the expanded form of the receptor binding domain of the various SARS-CoV-2 variant that has been reported from 2019 to 2020, and this can be seen here. It is difficult to see that. However, if one can notice this from the reference paper, they can see the mutations. For example, in the third row, there is a D is mutated to the Y in that variant, and if anyone is interested, they can go through the reference paper and check the high quality image of this uh, figure to understand where the mutation occurs in each variant. So this is just to give you an idea that major mutations are occurred in the receptor binding domain of the spike protein. And then they also compared whether this SARS-CoV-2 virus is comparable to the SARS-CoV and MERS-CoV or not, and they can find out that SARS-CoV-2 is comparable to the SARS-CoV virus. However, when they compare it with the MERS-CoV-2 virus, uh, they found around only 28% similarity, because there are around 51 amino acids that are different from the SARS-CoV-2 virus. And one of the example is can be seen in the bottom, sequence matching. In this, there is an additional amino acid that can be seen in the MERS-CoV-2 virus, whereas they are absent in the SARS-CoV-2 virus or SARS-CoV virus also. SARS-CoV sequence and SARS-CoV-2 sequence also. So this is just to provide you an overview that SARS-CoV-2 virus is similar to the SARS-CoV virus, and however it is different from the MERS-CoV virus. And let us come to our main topic, that is how this SARS-CoV-2 spike protein glycosylation varies with different cell system. In the previous video, we have compared three cell system, that is X cells, BIC2 cells, and BIC1 cells, and we compared the N glycosylation. However, in this we will compare the O glycosylation, and there they have only compared the two cell system. First one is the X cells, and another one is the BIC2 cells. So this is the circular figure in which the major O-glycosylation sites are shown, and from the legend we can understand that the inner circle represents the BIC2 O-glycans that are found in BIC2 sites, O-glycan sites in the BIC2s from the BIC2 cell system expressed spike protein, and the outer circle or the middle circle contains the O-glycan sites that are found in the spike protein that are expressed from HEC293 cell system. So, however, the complete details can be seen from the legends that what are the sites that are found in both the hex cells and spike protein expressed from hex cells and also from BIC2 cells, and it can be seen here. And this second and the right side of the panel, that is panel B, C, D, E, represents some of the highly abundant O-glycan sites, and compare it compare it between both the cell lines. As we can see for this panel B, we can see that this H1N1 is comparable in both the hex cells and the BIC2 express cells. BIC2 express spike protein, or the spike protein expressed from hex cells, at the T323 sites, and however there is some difference or in the case of N1 production at these sites. It is more in the case of HEC2 cells, whereas less is in the case of BSE2 cells expressed. And when we see the sites, that is 325 site, at 325 site, the H1N1 O glycosylation is more in the BIC2 cells, whereas it is very less in other HEC cell system. And whereas this N3F1 O glycan is more in the HEC cells, whereas it is almost negligible or at the reference level in the BIC2 cells. And in the case of S673 sites and T676 sites, we can see there is more variation that are observed in the case of HEC cells, as we can see that HEC cells show this S1, H3 and 2F1 expression, and also this H2 and 4F2 expression, and this H1N1 expression also, whereas the BSE2 cells shows only N1 expression, majorly shows only N1 expression, H1N1 expression and N3F1 expression. So it varies. The O glycosylation varies at the 673 sites, not only within the cell system but also across the cell system. And for this T676 sites, the similar trend observed as we have seen in the 673 sites, that is different glycosylation has been observed in the HEC cells, such as this S1, H3 and 2 F1 site, N3F1 site and H2 N2 sites. However, for BSE2 cell lines, the different glycosylation observed between N1 site, H1N1 site, N3F1 site, H2N1 and H1N2 sites also. So this represents this provides us an overall picture that not only just the N glycosylation changes within the spike protein of the SARS-CoV-2 when they are expressed in different cell system, but also the O glycan system. O glycosylation will also changes, as we can see from this result. And N glycosylation results we have seen in our previous videos. If you need to understand this study in detail, you can go through the reference paper that has been provided in the description, and in that paper you can also see the high resolution image of the previous figure, that is this figure, in order to understand where the mutation occurs in different variant. So that's all for this video. Thank you for your time.